Hello friends, this is Dr. Farheen and today's lecture is about introduction to medical microbiology. Medical microbiology. Before going to the series of lectures in medical microbiology, this is my first lecture on introduction. So in the introduction, the first and the foremost thing what I will be discussing is the meaning of the word microbiology, which you all must be very much familiar with. Then we will just refresh our known knowledge about little bit about the history of microbiology, and finally we will discuss what are the organisms that are included in the study of microbiology. What are the microbes that are to be studied? Now, first and the foremost, coming to the word microbiology. So friends, it's very easy to understand microbiology because microbiology is made up of two words, that is micro plus biology. Micro refers to small, and biology, as you all know, you have been studying this from your high school, the study of living organisms. Now, coming to the word micro, very easy to understand. Micro refers to small. We know it is small, but the question is how small? Is it small, say 1 mm, or 1 centimeter, or how much? Now here this small refers to very small enough to not to be seen by your naked eye. So scientifically speaking, it refers to scientifically speaking, it refers to 10 raised to minus 6 microns. Microns is equal to 10 raised to minus 6 meters. That we can say that 0 point followed by 6 zeros 1 meters. So this is the size of microbes which lies in this, and I'm so sorry. Size of this microbes is lying between say 1 micron to 15 microns, 40 microns, depending upon what it is. Now the organisms which are invisible to your eyes are measured in the microns, and these all belong to the microbe group, and this is what we study in microbiology. Now coming to so what is the meaning of the word microbiology? Micro refers to smaller organisms with a very small size, and biology refers to the living organisms, not the atomic or molecular structures, but the living organisms. Okay, now coming to the history, way back it was the first person who happened to come across microbes was Anton Van Leeuwenhoek. Anton Van Leeuwenhoek. Let me erase this so that you can be able to say it. Leeuwenhoek. Anton Van Leeuwenhoek. So what he did, he was able to see the microbes. He was actually a lens grinder, or you can say he was the one who used to grind lenses to see the threads of the clothes, and this lenses he happened to use it to see some material from his teeth, and he was able to see some moving structures which he named it as animalcules. So that was the start of microbiology. He got interested in these animalcules, and he started to draw diagrams of whatever he saw in the a plaque of the teeth, in his own semen, in the urine, in the feces, in the dirty water, whatever he could find, whatever he could feel like seeing, he would see under his simple microscope, or you can say a combination of lenses, and magnify them and draw the diagrams of them. And he at that time did not know much details about it, and he just named them as animalcule. He made a complete manual of the drawings, and he submitted to the scientific society, and at that time that was the end of it. but it was only after around 100 years when Otto Miller, who was a Danish biologist, he happened to work on the Leeuwenhoek's animalcules, and he gave them the names, and he placed them in the taxonomic category. Okay, that is a brief introduction. After that, there were many scientists who came after that, and there was first one was Louis Pasteur, who proved the theory of de novo synthesis, and then Robert Koch, who discovered anthrax, bacilli, so many bacteria, and culture techniques, so on. So it was a slow development, and then the era started with finding out different cells and different bacteria, viruses, so on, so on, so on. So this is not the lecture about the history of the medical microbiology, so we will not get into the details of it. Now I would like to mainly discuss the microbes. What are the microbes we are going to study in this lecture or in medical microbiology? Now, microbes included in microbiology. Now, for our convenience of understanding, I will divide this into two groups. What are those two groups? Number one, I will write it, I will name it as, say, acellular. I'm really very new to this technology, so please bear with me for some time until some videos where I can handle it more properly. Okay, anyways, now coming back to the microbes, I will classify them as acellular organisms, which do not have cellular they cannot be called as complete cell because they do not have the basic components of the cell. And the other one, I will name it as cellular ones. Now these acellular, I will put it in two. I will categorize them into two. One is the prions, the other one prions, and the other one the viruses. Coming to cellular, I will again categorize them into two. The prokaryotes and the eukaryotes. Okay, now coming to the prions, what are prions? Prions are nothing but units of proteins which are infectious, they can infect any human being, and they can cause diseases. They can spread the disease from one person to the other person, and hence they are infectious, and they do not have much, or you can say they have nothing in them except the proteins. They are just units of proteins which are infectious, capable of causing a disease. The second one is the virus. Now these are, I can tell you that these are little bit more evolved than the prions. Now evolution does not mean to the Darwin's evolution. Here I am just referring it to the structural content of the components. Now here in the viruses you have there is the genetic material, and there is one covering on that. That's it. It does not have a nucleus, it does not have a cell membrane, it does not have a cellular matrix, nothing. It just has a coat, you can say a cover, and within that is the genetic material. Now, okay, when we study the details, we are going to study what type of material it is and what are the different things and however. But here just I will give you a slight hint that this genetic material, either it can be RNA or it can be DNA, never the both. They both cannot be together in the virus. Either there will be a RNA or the DNA. Then this genetic material, or you can say nucleic acid, is covered by a cover which is known as the capsid. Now what is a capsid? This capsid is made up of protein basically, This is made up of protein basically. So you can say that viruses are just organisms which have genetic material covered with a protein capsule, or you can say protein capsid, sorry, not capsule, capsid. Okay, now the other one was, I referred to the cellular components, like say prokaryotes and eukaryotes. Now cellular, you know what is a cellular? Cellular means it should have at least the cell membrane. It should have a cell membrane. It should have a nucleus. And the cytoplasm, or you can say the cellular matrix, cytoplasm. Okay, these are the basic components which has to be present in the cell. Now these cellular components can be either classified as prokaryotes or eukaryotes. Pro yani primitive, eu yani developed. So karyo refers to the nucleus, karyo refers to nucleus. Now primitive, and you developed. Okay, now what is here now? What are prokaryotic cellular components? These are nothing but the organisms. These are the organisms with primitive nucleus. What is primitive nucleus? Nucleus which is not well developed. Yani, I can say without nuclear membrane. Okay, now coming to eukaryotes, the developed nucleus, they are with true nucleus. That means they have the nuclear membrane. They have the nuclear membrane, therefore they have well developed nucleus. Okay, so here let me give an example. Before that, I need to erase this so that I will be able to give you more details. Now let me just wipe it off so that I will give you a little bit details about eukaryotes and the prokaryotes. Okay, in the prokaryotes, mainly here we have the bacteria, a group of microbes. These are the bacteria which do not have a well-developed nuclei. They mainly lack the nuclear membrane. Hence, the genetic material is freely floating in the cytoplasm, just like this. If this is the bacterial cell wall or cell membrane, within this, the nucleus or the nuclear material is just lying freely in the cytoplasm. This is the cytoplasm. Okay. Now in the eukaryotes, there are many categories. Mainly here I would like to refer, in case of microbiology we study, is the fungi and the parasites. Further, the classification of fungi and parasites we will discuss one by one when we go into each one of the chapter. So friends, what we learned today in this video is, let me just put it down in a neater way so that you will be able to remember this. You will be you can make notes whenever I'm online, or you can just have my notes which I will be giving in the description box later. Okay, now mainly what we studied was microbes. These are into two: acellular and cellular. Acellular, I had two classifications. One is prions and the other one was virus. Here in cellular, again two. Number one was prokaryote and number two was eukaryote. And prokaryote, we have only one group here, that is the bacteria. And in eukaryote we had two, that is the fungi and the parasites. So that's all for today, this lecture, guys. Please subscribe my channel and click the like button for encouraging me for preparing more lectures and uploading more videos. And do subscribe it so that you can get notified for next uploaded lectures on medical microbiology. Thank you.